Hello, everyone. First of all, I'd like to thank to the organizing committee for giving me the opportunity to present my work here. Before I begin, let me briefly introduce myself. My name is Dr. Irem Gulfem. I've been working as a research assistant in Üsküdar University, Department of Molecular Biology and Genetics for about 7 years now. Today, within the scope of this conference, I'd like to present a study based on my PhD thesis. The title of my presentation is Investigating the Effect of NHE6 Gene Modulation in Alzheimer's-Related Gene Expression Profiles. At this point, before I begin with my presentation, I would also like to thank to my PhD thesis advisor, my supervisor, Professor Dr. Musin Konuk, for his valuable guidance, support, and contributions throughout this study. This thesis project was supported by the Scientific and Technological Research Council of Turkey, TÜBİTAK. And in today's presentation, I will first provide a brief background on Alzheimer's disease, including its major pathological and genetic features. Then I will focus on the endolysosomal system, intracellular pH regulation, and sodium proton exchangers, especially NHE6, which is encoded by the SLC9A6 gene. After that, I will explain the aim of the study, the experimental design, and the main methodological steps. And finally, I will present the results obtained from gene expression analysis, ELISA measurements, and discuss the possible implications of this gene, of this protein, NHE6 modulation in the context of Alzheimer's disease-related molecular pathways. I will now briefly explain the background of Alzheimer's disease, the relevance of endosomal pH regulation, and why NHE6 was selected as the main focus of that study. Alzheimer's disease accounts for approximately 60 to 80% of dementia cases, making it the most common cause of dementia worldwide. The disease particularly affects brain regions that are critical for learning and memory. Among these, the medial temporal lobe, including the hippocampal formation and neocortical structures, are highly vulnerable. Pathologically, Alzheimer's disease is mainly characterized by two features. First is the accumulation of extracellular amyloid plaques, which are primarily composed of amyloid beta peptides. The second is the formation of intracellular neurofibrillary tangles, which are mainly composed of hyperphosphorylated tau protein. As shown in the slide on the right side, when we compare a healthy brain with a brain affected by Alzheimer's, we observe neuronal loss, synaptic degeneration, and structural atrophy. These changes are directly associated with the progressive cognitive symptoms observed in the patients. Importantly, the number of affected individuals worldwide is expected to exceed 100 million by 2050, which emphasizes the urgent need to get a better understanding of the molecular mechanisms underlying this disease. The pathophysiology of Alzheimer's disease is complex and multifactorial. One of the central pathological features is the formation of amyloid plaques. These are extracellular deposits of amyloid beta protein, and they may appear in different morphological forms depending on the stage and severity of the disease. Another key feature is the neurofibrillary tangles. These tangles are composed of abnormal filaments of hyperphosphorylated tau protein. Under physiological conditions, tau plays an important role in microtubule stabilization. However, when tau becomes abnormally phosphorylated, it loses its normal function and it contributes to neuronal dysfunction. The third one you see in the list in here in the slide is synaptic loss. It's another major component. The synaptic damage is one of the strongest correlates of cognitive decline in Alzheimer's disease. Memory impairment often appears in early stages due to the synaptic degeneration in the neocortex and limbic system. And finally, oxidative stress also contributes neurodegeneration. It's associated with amyloid beta production and accumulation, microglial activation, and mitochondrial dysfunction. So the Alzheimer's disease should not be considered only as an amyloid-related disorder. Rather, it's a network of interconnected pathological mechanisms. Genetic factors also play a very important role in Alzheimer's disease. It's been reported that approximately 70% of Alzheimer's disease cases are associated with genetic factors. In particular, most cases of early onset Alzheimer's disease show autosomal dominant inheritance. Mutations in APP, PSEN1, presenilin-1, presenilin-2, and ApoE genes found to be associated with Alzheimer's disease. They are these diseases that I mentioned, they are directly involved in amyloid precursor protein processing and amyloid beta generation. On the other hand, ApoE, particularly the ApoE4 actually, is considered one of the strongest genetic risk factors for late onset Alzheimer's disease. At this point, I'd like to move to the endolysosomal system because this system is closely related to both intracellular trafficking and Alzheimer's disease pathology. The endolysosomal system consists of several intracellular membrane-bound compartments, including early endosomes, late endosomes, and lysosomes. These compartments are dynamic structures and can interconvert with one another. One of the most important functions of the system is to create and maintain an acidic intracellular environment. This acidic environment is necessary for protein degradation, receptor recycling, vesicular trafficking, and many other things. The pH of different cellular compartments is tightly regulated. While the cytoplasmic pH is approximately 7, the pH of the endosomal and lysosomal compartments are more acidic, somewhere around 6.5 to 4.5. Even small disturbances, even small changes in endosomal or lysosomal pH may affect protein processing, receptor signaling, and vesicle trafficking. Sodium proton exchangers, also known as NHEs, they are very important regulators of cellular pH. They mediate the exchange of sodium ions and protons across cellular membranes. They contribute to pH homeostasis, cell volume regulation, ion balance, and intracellular signaling. NHE proteins are localized not only at the plasma membrane, but also within the intracellular organelles. Among the NHE family members, NHE6 has attracted attention due to its localization in early endosomes and its potential role in neuronal function. Now in here, I'd like to focus more on NHE6 because it's the main focus of our research too. NHE6 is encoded by SLC9A6 gene, which is located on the long arm of the X chromosome. NHE6 is considered as a key regulator of early endosomal pH in neurons, especially in hippocampal pyramidal neurons. NHE6 is widely distributed along axons, the soma, and dendrites. It also shows substantial colocalization with endosomes in dendritic spines. This is important because dendritic spines are highly active regions involved in synaptic transmission and plasticity. Furthermore, NHE6 shows strong colocalization with both presynaptic and postsynaptic markers, including synaptic vesicle protein 2 and postsynaptic density protein 95. This suggests that NHE6 may not only regulate endosomal pH, but may also contribute directly to synaptic transmission, neuronal signaling, and possibly neurodegenerative mechanisms. This table summarizes the relationship between NHE6 and endosomal pH regulation. In early endosomes, NHE6 contributes to the balance between proton accumulation and proton leakage. By exchanging sodium ions and protons, it prevents excessive acidification of the endosomal lumen. When NHE6 function or expression changes, endosomal pH may also change. This can influence vesicular trafficking, receptor recycling, and protein processing. In the context of Alzheimer's disease, this is especially relevant because amyloid precursor protein processing and amyloid beta generation are closely linked to intracellular trafficking pathways and endosomal compartments. Therefore, modulation of NHE6 may indirectly or directly affect Alzheimer's disease-related molecular pathways. So now I'm gonna show which target genes we choose related to Alzheimer's. We selected several target genes associated with Alzheimer's disease pathways. Here on the right, you can see a pathway map. You can find the positions and biological relevance of these genes. We evaluated it using the CAC pathway analysis database. The selected target genes were APP, presenilin-1, IRS1, and CKY epsilon. The first target gene, APP gene, it encodes amyloid precursor protein. APP is central to Alzheimer's disease pathology because the amyloid beta peptides are produced through the proteolytic processing of APP. The second target gene is presenilin-1, which encodes presenilin-1 protein. This is a core component of the gamma secretase complex. Certain mutations in this gene cause familial Alzheimer's disease. These mutations may alter amyloid beta production by reducing shorter amyloid beta peptides, such as amyloid beta 40, or increasing the longer and more aggregation-prone peptides, such as amyloid beta 42. The third target gene is IRS1, insulin receptor substrate 1 gene. This protein transmits signal from the extracellular environment to the intracellular environment and coordinate multiple cellular processes. In Alzheimer's disease, impaired insulin signaling has been associated with neuronal dysfunction, altered metabolism, and cognitive decline. Our last target gene is CKI epsilon, or casein kinase 1 isoform epsilon. This gene is involved in several cellular processes, including DNA replication and DNA repair. More importantly for this study, these family members have been associated with tau-related pathology and neurofibrillary tangles in Alzheimer's disease. Thus, by analyzing APP, PSEN1, IRS1, and CKY epsilon genes, we aim to evaluate whether the modulation of SLC9A6 expression could influence different Alzheimer's disease-related molecular pathways. So the main aim of this study was to investigate the effects of increased and decreased expression of SLC9A6 gene, which encodes NHE6 protein, on Alzheimer's disease-related genes. For this purpose, we used the human neuroblastoma cell line, SHSY5Y cell line. We specifically analyzed the effects of SLC9A6 modulation on the expression levels of APP, PSEN1, IRS1, and CKY epsilon. In another words, actually, we asked the question like, can changes in NHE6 levels influence molecular pathways associated with Alzheimer's disease? By answering this question, we aim to better understand the relationship between early endosomal pH regulation and Alzheimer's disease-related gene expression profiles. Now I will move on to the materials and methods section. The experimental workflow included several steps. First, we optimized the cell culture conditions and generated a cell proliferation curve so we can decide when give the amyloid beta to the cells, when to subculture them. Then we performed cell viability assays to determine the proper amyloid beta treatment concentration and exposure time. After that, the cells were differentiated into a more neuron-like phenotype. The Alzheimer's disease model was established to that neuron-like cell lines, differentiated cell lines, using an amyloid beta 142 peptide. Then our target gene, SLC9A6 gene expression, was expressed using siRNA and upregulated at the same time using a vector mediated overexpression. Following these manipulations, NHE6 protein levels were measured by ELISA to prove that we see an increase and a decrease in protein level 2. Then total RNA was isolated. We checked the RNA quality, cDNA synthesis, complementary DNA synthesis, and gene expression analysis with real-time PCR performed. Finally, amyloid beta 142 levels were measured before and after the manipulation, and statistical analysis were performed. For cell culture optimization, the cells were maintained under proper culture conditions. Cell counting was performed using a TOMA counting chamber. You can see a picture of it on the right side. The cell concentration was calculated using the formula shown here. And the image on the left represents how the cells are seen under the microscope right before the cell passaging, subculturing. Since the SHSY5Y cells are neuroblastoma-derived cells, they require differentiation to better represent the neuronal characteristics. To do that, neuronal differentiation was performed using a protocol based on retinoic acid and BDNF treatment. You can see the workflow in here, how differentiation is made. To confirm the differentiation, we checked them under the microscope too, of course, but we checked the gene expression levels too, neuronal differentiation markers. You can see the name of the genes and primer sequences shown in the table. By evaluating these differentiation markers, we aimed to confirm that the cells acquired neuronal features before moving on with the other steps. After neuronal differentiation was successful, we started to create the Alzheimer's disease model. To do this, we used synthetic amyloid beta 142 peptide. The peptide was dissolved in DMSO and then diluted with PBS and prepared a stock solution. Then this stock solution is incubated at 37 degrees Celsius for 3 days and mixed daily by pipetting. So we can see some aggregation formation inside. Based on the cell viability assay results and previous literature, we decided to treat the cells with 10 micromolar amylate beta 142 for 48 hours. In this slide, you can see the experimental groups that we worked with, the abbreviation of each group and characteristics. In each step, we also had a control group, so we can just check if we do the differentiation correctly, if we performed the disease model correctly. So in each step, we had a control group for that too. And also, as you can see from the workflow on the right, after this, first establishing the cell culture, then neuronal differentiation, Alzheimer's disease model, then our study divided into two. So with one group, we decreased the expression of NHE6. In the other group, we increased the expression of NHE6. Then also we have another group that's not showing in here, a negative control group. We didn't do anything to them, so we had chance to compare the gene expression levels at each step. For the suppression of SLC9A6 gene expression, we used a specific siRNA targeting the transcript. The targeted gene regulation is marked in green on the genomic map shown on this slide, and transfection was performed using a transfection reagent. In addition to gene suppression, we also performed vector-mediated overexpression of our gene. For this purpose, the vector we used, shown in here, containing the cloned sequence information of the SLC9A6 gene, was obtained from AdGene. Transfection was performed using DNA fectin plus transfection reagent. By comparing the siRNA group, the vector group, and the control Alzheimer's disease model group, we aim to evaluate the bidirectional effects of SLC9A6 modulation. After gene expression manipulation, NHE6 protein levels was determined using ELISA. This was important because the changes at the mRNA level do not always directly reflect the changes at protein level. Therefore, measuring the NHE6 protein levels provided an additional confirmation of the biological effect of SLC9A6 gene manipulation. I mentioned two our target genes and also the gene that we manipulated. It's on the first row. So we performed gene expression analysis with them using real-time PCR. You can see the information about the genes, the oligonucleotide sequence, and some primary informations in here. So with all of them, we performed real-time PCR analysis too. Now I'm going to present the main results of our study. This curve was generated to understand the growth kinetics of the cells and to determine the proper timing for experimental interventions. Next, we performed MTT assays, cell viability assays, to determine the most proper amylate beta 142 treatment dose and exposure time. Different concentrations and time points were evaluated. Based on the MTT assay results, the most suitable treatment condition was determined as 10 micromolar amylate beta 142 for 48 hours. This condition was selected because it was sufficient to induce a disease-related cellular stress response while maintaining enough cell viability for downstream molecular analysis. This slide shows the results of neuronal differentiation and the establishment of the Alzheimer's disease model. During the differentiation process, morphological changes were observed in SY55Y cells. The cells gradually developed more neuron-like features, including elongation process. In addition to microscopic observation, neuronal differentiation was evaluated by gene expression analysis of differentiation biomarkers 2, MAP2 and NAFL genes. The results supported the successful differentiation of SH-SY5Y cells before amyloid beta treatment and gene manipulation experiments. After establishing the differentiated Alzheimer's disease model, we manipulated. Also, before I move, I have to mention that after this differentiation, and after we prove it, then based on the literature, based on the method from the literature, we gave the decided amount of amyloid beta 1.42 to the cells to induce the Alzheimer's disease model. And after establishing the differentiated Alzheimer's disease model, we manipulated the SLC9A6 gene expression using siRNA and vector. Compared with, if we compare these three groups together, the first group you see in here, they are just differentiated and Alzheimer's induced. And this group is the group that we applied siRNA, and this group is group that we apply vector. So in the manner of SLC9A6 gene, you can see in the group that we applied siRNA, we see a decrease in the gene expression. And in the vector group, we see an increase of gene expression when we compare it with the negative group. So we can say that these results indicate that the experimental manipulation of SLC9A6 gene was successful. Following the gene expression analysis, we measured intracellular NHA6 protein levels by ELISA. And in here you can see our results. On the left, this is the standard curve we used, and on the right, you see our results. The ELISA results allowed us to evaluate whether the changes in gene expression were reflected at the protein level. This step was especially important because NHE6 is the functional protein product of this SLC9A6 gene, and its level may directly affect endosomal pH regulation. And by checking the protein levels, you can see this is our negative group, this is the vector group, we see an increase. And our siRNA group, we see a decrease in protein level too. So we proved manipulation in protein level as well. After we made them all, then we analyzed the Alzheimer's disease-related gene expression profiles by real-time PCR. Here you can see the results of APP gene expression. It's found to be reduced 5.2-fold compared to the negative group in siRNA applied group. In the vector group, on the other hand, there wasn't a significant difference. When we check our second target gene, presenilin-1, again with siRNA group, we see a significant decrease on gene expression level, 6.9-fold compared to the negative group. But with vector group, the results were not significant. Our third target gene, IRS1 gene expression, also reduced 17.33-fold compared to the negative group in the group that we applied siRNA. On the other hand, similar to the other groups, there was no significant difference with the group that we applied vector. This is our last target gene, it was CKY epsilon. In the SLC9A6 suppressed group, siRNA group, gene expression was reduced to 0.7-fold compared with the control group. As you can see here in this graphic, again, with the vector group, there wasn't a significant difference. In addition to all this gene expression analysis, we also measured amyloid beta 142 protein levels by ELISA-2, both in intracellular and extracellular places. So in both of them, intracellular and extracellular, amyloid beta 142 levels were measured. The standard curve that we generated is on the left. And on the right, you can see our measurements and our statistical analysis results too. The most important observation was that following SLC9A6 suppression, intracellular amyloid beta 142 levels were reduced. This result supports the idea that NHE6 may be involved in the regulation of amyloid beta accumulation, possibly through its role in endosomal pH and intracellular trafficking. Now I'd like to discuss the importance of these findings. Sodium proton exchangers have a very high ion transport capacity. It has been reported that these proteins can transport approximately 1,500 ions per second. It is a very high amount. Therefore, even relatively small changes in the expression of these transporters may have functionally important consequences. In our study, siRNA-mediated suppression of SLC9A6 gene significantly reduced the expression of Alzheimer's disease-related genes, including APP, presenilin-1, RRS1, and CKI epsilon genes. In addition, following the decrease in SLC9A6 levels, intracellular amyloid beta 142 levels were statistically and significantly reduced by 0.31-fold, with a coefficient of variation below 20%. Our findings are also consistent with previous studies showing that NHE inhibition may have neuroprotective effects in experimental models. In animal studies, NHE inhibitors have been shown to reduce cellular damage by limiting intracellular sodium and calcium accumulation. This may reduce exocytotoxicity-related injury and downstream cellular stress responses. NHE inhibition has also been associated with the suppression of microglial activation. In particular, neuroprotective effects of NHE inhibition have been observed in hippocampal CA1 pyramidal neurons. Some studies have also shown improved functional recovery following NHE inhibition. There is also literature specifically supporting the relationship between NHE6 and Alzheimer's disease-related mechanisms. Previous studies have shown that SLC9A6 deficiency can significantly reduce amyloid plaque accumulation. Moreover, ApoE4-induced endosomal trafficking defects and increased amyloid plaque accumulation can be corrected through inhibition of NHE6 protein. This is particularly important because ApoE4 is one of the strongest genetic risk factors for late-onset Alzheimer's disease, as we mentioned. These findings suggest that NHE6-specific inhibitors may represent a potential therapeutic strategy for Alzheimer's disease. In this context, our findings provide additional support for the hypothesis that NHE6 SLC9A6 may be a relevant molecular target in Alzheimer's disease-related cellular pathways. Now I will summarize the main conclusions of our study. As a result of this study, we found that inhibition of NHE6 SLC9A6 reduced intracellular amyloid beta 142 protein accumulation. These findings suggest that regulation of early endosomal pH may have a direct effect on amyloid beta metabolism. In addition, following the SLC9A6 inhibition, we observed a decrease in the expression of Alzheimer's disease-related genes, which are listed in here. These findings suggest that NHE6 inhibition may not only reduce amyloid beta accumulation, but may also regulate molecular signaling pathways involved in Alzheimer's disease. However, it is important to emphasize that this study was conducted in an in vitro cellular model. Therefore, further studies are needed to validate these findings in more complex biological systems, including animal models too, and clinical studies. In future research, pharmaceutical agents targeting NHE6, SLC9A6 inhibition should be investigated at preclinical and clinical levels. Such studies may contribute to the development of innovative mechanism-based therapeutic approaches for Alzheimer's disease. The references supporting the background and discussion points are listed in here. Thank you very much for your attention.